let's okay. Whenever we plan the investigation, first of all, we need to know what we are doing, or maybe what is our plan, what is our aim, what problem we are addressing. Remember that in biology, the problem does not mean as same as the problem in the English. In English, we often say the problem is something that is bothering us, something that is out of our daily routine, something that is interfering with our daily work. That is a problem. But in biology, the problem appears to have another meaning, and uh, that is some sort of scientific issue, scientific research, scientific plan. And for every scientific plan, we should have a very important thing, that is a hypothesis, although hypothesis is not there in our syllabus. But hypothesis is somewhat like, if I suppose, if I say that if light is a limiting factor, if light is a limiting factor for or photosynthesis, then changing the light, changing the light intensity would would result in change in rate of photosynthesis. That is what we think about in as a hypothesis. But obviously, we do not have to design the hypothesis at this level. After the hypothesis, there are two things in this particular investigation. I guess you can see the word change. Can you see the word change there in the hypothesis statement?
Yeah.
When something's changing, when something's changing, or when the rate is changing, uh, rate of something, when rate of something is changing, then we say that thing is a variable. Suppose in the statement below, what do you think is changing?
The light,
And
the rate of photosynthesis.
rate of photosynthesis, yes. So two things are clearly changing in the rate of in this experiment. So we would say we have two variables. One is one variable is called as light, and other variable is called as rate of photosynthesis. Both of these are changing their values. Rate of photosynthesis. Both of them are changing their values. So every factor, every factor that would change its value, every factor that would change its value would be called as a variable. Would be called as a variable. So now, but hold on. Over here, you can see that one of these factors is changed by us? Can you tell me which factor is being changed by us?
The light.
Light. The light is being changed by us. Light is variating by us. In this case, light is called as independent variable. Any factor, any variable that is being changed by us would be called as the independent variable. Any of those. Like if there is any experiment in which you are changing the temperature, in that case, the independent variable would be the temperature. So independent is the one for which we are independent to change. We are independent to change its value. That would be called as independent variable. Is it understood?
Yeah.
Okay. Next is why the rate of photosynthesis is changing.
Because we are changing light.
Because we are changing light. Or in other words, you can say because we are changing the independent variable,
Yes.
a factor that changes as an effect of as an effect of independent variable independent variable is called as dependent variable dependent variable, which means that dependent is depending upon independent. Dependent is depending upon independent. When you are changing the independent, as a result, the dependent is being changed. Is it understandable?
Yes.
So in this case, what is the dependent variable?
The
The
rate of
rate of
photosynthesis.
photos. Yes. So it means light is being changed by us while the photosynthesis is changing because the photosynthesis is depending upon the independent variable, that is light. So that is why we call that the dependent variable. Basically, you can, in other words, you can say that the dependent variable is our result. This variable is our result. And it is quite obvious because when we change the light, that would change the rate of photosynthesis, and that would ultimately lead to the change in the rate of that independently would change to in the value of the dependent variable. That is the whole phenomena. That is the whole concept of independent and dependent. Okay, is it clear?
Yes.
Okay. Now, independent, dependent, these two variables, they work like they are important. If you want to do an investigation, in every investigation, we need to we need to uh, keep one independent variable at least. Variable at least. In O level or IGCSE biology, we only keep one independent variable, but sometime in many other investigations we can keep more than one independent variable. Okay, but uh, another very important line I'm about to write, specifying specifying the variables specifying the variables, or let me change the sentence. I would say specifying the dependent and independent variable, dependent and independent, does not mean that there are no other variables. Does not mean that there are no other variables. Now, what does this mean? There are some other variables. There are some other factors that can change their value, that can change their value, but we don't let them. But we don't let them change. We don't let them change. Those particular variables are called as constant variables, constant variables, or in other words, they are also called as standardized variables. What does this mean? Well, once more, variable. That clearly means something that can vary.
Yeah.
Now, constant variable means that these factors can literally change the results, but we are not letting these factors to interfere, and we are not letting these factors to act. That is the meaning of constant variables. We are keeping them constant. We are keeping them in line. Strictly we are strictly not letting them change. That is the meaning of constant variables. Suppose uh, we just assume that the light is changing the rate of photosynthesis. Did we?
Yes.
But is there any factor that can also affect the rate of photosynthesis?
Like you mean other factors?
Yes.
Um, yeah, there are other factors.
What other factor can change?
Um, so temperature,
Hmm.
carbon dioxide concentration.
Temperature or carbon dioxide. Now, these factors, if light is independent variable, then in that case the other two the other two have to be standardized. The other two means the temperature and carbon dioxide have to be standardized. Why do you think is that? Why do we need to standardize the temperature and carbon dioxide if the light is independent? Hmm?
Sorry, I didn't get that wording. So can you repeat that?
If we are making the light as an independent variable,
Yeah.
then why do we need to keep the temperature and carbon dioxide constant or standardized?
Because the variable that we're testing is the light, right? Not the carbon dioxide
Yes.
or the temperature.
First conclusion is the independent has to be strictly followed. First thing, it has to be strictly followed. Number two, we won't be able to figure out that what factor what factor is affecting the dependent variable. And number three, it would disturb our results. It would disturb our results. Or you can also say accuracy. So that is why some factors have to be standardized or some factors have to be very much constant. That is the reason. Clear?
Yeah.
Okay. Now, next is uh, when we have seen what is independent, what is dependent, what is constant. Like we have particularly checked that what are independent and dependent. Now, what other things we should be we should be knowing very well in in an investigation. Okay, one of those factors is the apparatus. One of those factors is the apparatus and how it works. The apparatus and its working. The second thing we should know is the chemicals and their use, chemicals and reagents. Even you can include indicators in this list, indicators. And like if you want to know where can we see the independent variables, I'm sorry, where can we see the apparatus, then I can open a file in front of you that would help you understand that where can you find the apparatus. Mm, oops, there must be somewhere here. Yeah, yeah, this is. Read all these carefully and then tell me.
Okay, yeah, finished reading.
Okay, now many factors you are seeing here, many apparatus. The important thing is we should be knowing all their names, first thing. And the second thing is we should be knowing that how they work. That is the major point of showing you the list. Can you hear me?
Yep.
Okay. So let's say, first of all, they are saying balance to measure beakers of various size, bungs, filter funnels, filter paper, forceps. Now they may ask you sometime, they may ask you that what out of all these factors, or maybe there is a question, there is a very, very particular question in the past papers, and you will find that a question in P6 that they may ask you, you need to, or how a student would measure the exact volume of this particular solution to perform the experiment. So now it depends upon the total volume. Suppose the total volume is 50 ml. You know what would you use?
If it's 50 ml,
Hmm.
um, I think you would use the cylinder, right?
Measuring cylinder, exactly, exactly. Then if it's only 10 ml or 5 ml, you may use the pipette. If it is the 2 ml, if it is only 2 ml or 1 ml, you may use the syringes, graded syringe. And if it is drops, then you can use the droppers.
Yeah.
So this list that is provided over here, you can easily read it and figure out that what you need to know, how these things would work, how apparatus would work, and how you should be answering that what would be used over here. Clear?
Yeah.
Okay. Then moving above, there are chemicals. So let's read these chemicals and tell me.
Yeah. Finish reading.
Now, you may have read some of the reagents, and also you may have read some of the uh, chemicals, other chemicals like ethanol. You may have read the water. You may have read DCPIP over here. So all these basically are there to know to know that what are about to be what chemicals you'll be using in these experiments. So suppose you'll be reading over here hydrogen carbonate indicator. Yeah, hydrogen carbonate indicator. You know, this hydrogen carbonate indicator basically is used when you want to know about this hydrogen. This is the hydrogen carbonate. When you want to know about the amount of carbon dioxide in a solution. Like if the carbon dioxide, it has three colors. One is original, one is original, then another color when the carbon dioxide is high. When the carbon dioxide is high in solution, the solution becomes acidic. So it would change into that color, relevant color, when it is acidic. And if it is alkaline, it means that the carbon dioxide is less CO2. In case of less CO2, I would like you to, okay, let me open that document that I shared with you about those chemical tests about those chemical tests. Just look at here. Carbon dioxide indicator is hydrogen carbonate. Its original color is red. If the solution has more carbon dioxide, it means it would have decreased pH. It would turn yellow. And if it has more carbon dioxide, sorry, if it has less carbon dioxide, which means alkaline or increased pH, it would turn purple. So that is the meaning of reagent. Then some of the chemicals are mentioned over here. This document is going to help you a lot in all these experiments. Okay. hydrogen peroxide, and then all these, then we will be knowing about every each and every. So this is the chemicals and apparatus for an investigation. Understood?
Yeah,
I
understood.
could not hear you.
Sorry, I was on
Okay,
mute. I
you
was
might.
on mute.
Okay, so next thing in every experiment is the once you have done all this, yes, then presentation of data. Presentation. A presentation of data can be done in two ways. One is tabulation. In tabulation, uh, when you would try to make a table, then you should be arranging your independent variables, independent values or variables in vertically, and then against it, you should be arranging your dependent variables, dependent values. That if we change this, then dependent values would be appearing as a result of it. Clear? So when we'll be learning about how the table is designed, I'll tell you that again. Another way of presenting the data is graphical. You would be making the graph. Now, this is very important. In every graph, first of all, you must have a very good knowledge about the axes and divisions of boxes. And I guess this is the thing of junior classes. Divisions of boxes. Another very important thing about the graph is the where would you put independent and where would you present, at which axis you would present the dependent variable.
Um,
So, do you have idea?
on a graph, uh, I think it's x-axis, right, for independent variable.
Yes, 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 yes. At x-axis, you would be mentioning the independent variable. And obviously, at y, you would be presenting the dependent. And at the end, a very typical question would be asked. Describe what is happening in the graph. One of the most typical question in all P6. And many times they would ask, explain. So obviously, when you would see the graph, you would be able to describe. And uh, when you would when you have described the graph, then after that, you would be explaining it on the basis of your knowledge. Understood?
Yeah.
Hmm. Once that is done, now you are there for conclusion. See, there are two things. One, the result is something else, and conclusion is something else. Result is on the basis of data, what changes are happening, what changes. And conclusion is you justify those changes, why it is happening. These two things are different. So always remember that concluding it would mean that you would have to present the conclusion on the basis of your knowledge and your understanding of independent variable and its effect on dependent one. Next, now next two terminologies are a bit weird that I am about to explain. Yeah. Uh, what are the accuracy and precisions? The accuracy and precision are actually, what is the meaning of these two things? Remember, accuracy of observations and data. How your data is accurate, how much your data is accurate. Like you are going to improve the accuracy of your data on the basis of repeated experiments. On the basis of repeated experiments, that your values are accurate, your values are exact. Got it?
Yeah.
Accuracy is related to the data always. What are your measured values? Then comes precision. Taking reading from apparatus. Precision always related to how you would be observing the readings, like observing or using the apparatus. That is called as precision. So the accuracy, they may ask you that how can you make your experiment more accurate? Obviously, when you are going to take when you are going to make your experiment more accurate, the only and one of the most important thing is repeat your experiments. Once you repeat your experiments, your this is also called as replication of the experiments. So that would be more accurate results. Comparison. There are two answers to this question. How would you make it accurate? First, replicate. Then keep the distance between the values shorter. Between the values shorter. Now, what does this mean? Let me tell you. This means that if you are working on enzymes and you are assessing at 0 degrees centigrade, then 15 degrees centigrade, then 30, then 45, and then 60. To be more accurate, you should be reducing the distance 0, 10, 20, 30, 40, 50, etc. That is called as your accuracy, repeating the experiment. That is how you can get the accurate readings. Observing it, taking the reading from the scale. Like you are looking at the scale and you have made a human error by mistakenly looking at the wrong measurement. That is the thing of the precision. So this is the basic aim. These are the basic aims of accuracy and precision. Understood?
Yeah.
Okay. And in the end, you are supposed to be in P6. Now, this is about a whole experiment that is almost of 22 marks. Most of the time, 22 marks, 20 to 22 marks are present in, are basically divided into one experiment. Then there is like, this is very important lesson you must know. And this type of lesson is most of the students, they ask for that that these things should be explained in only one lesson. Then in P6, you may be asked about to plan an investigation. You would be provided with the hypothesis. Suppose effect of temperature on the enzyme action. Now, to plan an investigation, you should be following the same method. First of all, mention the independent variable, mention the dependent variable, mentioning the apparatus, the chemicals, the way of doing, that's all. You don't need to mention any any particular data. You don't need to mention any particular conclusion. No, 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 no. Most important thing about every investigation that you would plan in the CIEs would have a very clear instruction, and that is the investigation should be written in such a clear way that it must be followed by another person. Like, you should be mentioning them so clearly, so accurately, that another person should be able to follow them.
Yeah.
That would be a six marker. That would be a six marker. And that is basically the one you were asking for. In I did not know that in physics and chemistry that is of 9 marks, but in
It
biology.
varies between 6 and 9. Mm.
Okay, okay. So in biology, if you look in the if you find in the past papers, it may varies between 6 to 7 marks. It necessarily would be six marker, but sometime it can be exceed to 7 marks. But this is one of the most easiest question. You know why? Because the pattern of all these questions is same to same. What? Independent variable, dependent variable, apparatus, chemicals, way to use, hazards, precautions. That's all. Fine. So this is one of the most easiest question. I forgot to tell you about the precautions, but you are smart enough. You may know what are the precautions in the experiments. Like, if you are doing an experiment in which you are using you are basically assessing the effect of temperature, then your precaution should be keep yourself away from the heated objects, something like that. So that would be your precaution, and many other things. Clear?
Yeah.
Okay, did I tell you about the water bath before? What is a water bath?
Um, a water bath is basically a tub of water, right? That you usually put. Um, I don't know what you use it for, but yeah, I know what you're talking about.
Water bath, basically, you are not very far. The water bath is basically used when you are doing an experiment in which you don't want to heat your apparatus directly. You don't want to heat your objects directly. Instead, you heat the water that would ultimately heat your objects, your apparatus. So what do? You heat the water. There are two ways to heat the water. One is the mechanical. You would use the machinery. You would use the machinery, but in the water bath you have to keep the temperature constant always. To keep the temperature constant always. So in the machine water bath, you have a thermostat like an air conditioner. What happens in the air conditioner when the temperature gets to a certain point, it trips. Is that right?
Yeah.
Similarly, in the machine water bath, the when the temperature of the water would rise to up to a certain level, it would trip. But we sometimes make a water bath of our own. How? We fill up a beaker up to the half with the water, and then we put our beaker into that water. Now we would be heating the water, but we have to keep the temperature constant with a very careful reading of the thermometer. So when we make a water bath of our own, that makes something very you can say sometime that is a source of error. That is a source of error. Clear?
Yeah.
Now these are the chapters in which you are supposed to be finding out some of the experiments. But first of all, how would you be able to find out the experiments? That which experiments are in your syllabus? Let me guide you. When you would open your syllabus, you would have a very clear instruction. Let me open the chapter that we just have finished. Suppose there is, uh, yeah, look at this. Transpiration. Investigate and describe the effect of variation of temperature and wind speed on the transpiration rate. Wherever you would find the word investigate, it means it is purely related to your P6. P6 can have variable questions, always remember. But one of the question would always be from the investigations like these. They would be mentioning investigate and describe. And when they would be mentioning that, you should be able to understand that what they mean. They mean that you need to investigate according to the independent and dependent variables. Suppose that in your book they are using this particular apparatus called as, this apparatus is called as photometer. This apparatus is called as photometer. Oh, using the forgot to mention. This photometer would be available, and in this particular experiments, you may use the wind or temperature to test that whether the transpiration would increase or decrease. So if you are using the temperature to check whether the transpiration would increase or decrease, what would be your independent variable?
Um, in this specific one, right?
Yeah, using the temperature.
So you're changing the temperature.
If you are
So the
changing the
changer,
temperature,
if you're changing the temperature, then it's the independent variable.
it is independent variable, yes. So if you are changing the wind, obviously it would be the wind would be the independent variable. And in this experiment, the photometer one, basically what do we often do? You see, this is a very strange, you can say, pipe-like thing. Very strange pipe-like thing. And on one end, you place the branch. On one end, you place the branch, and on the other end, you put that particular uh, end of tube into a beaker of water. When you would put a plant into the rubber tubing, you would be putting the wax around it so that no water is further lost, and you would see the position, the movement of the bubble. The movement of the bubble. The movement of the bubble in the given time would give you the idea that what is the transpiration rate, because the higher the transpiration, the faster would be the movement of the bubble towards the left. Is that understood?
Yeah.
So that is why we use the air bubble. And why is a reservoir? Because once the water bubble has moved to the left, for the next reading, we need to move it back to the zero. So we put more water by opening the reservoir. And when the reservoir is opened, we can easily push the water bubble, or sorry, air bubble back to the point zero where it would start again.
Okay.
This is one of the experiments that we do. There may be some other experiments related to the photosynthesis. Yes, in the photosynthesis, we also do some experiments in which we basically uh, plan to change the rate of photosynthesis by putting the light closer to the plant or away from the plant. So I'll tell you that, but before that, let's continue some of the previous topic. I would give you a 2 minutes break. Just analyze the whole document that I have written today and see if you want to know anything more. Got it? 2 more minutes.
I've been viewing it from your screen. I don't have the link.
I did give you the link yesterday, or maybe day before yesterday.
I forgot to save it.
That's not good. Keep the links, or do you have all the links saved with you?
Yeah, I have the previous one.
Don't lose these links. These are important.
Okay, yeah, finished.
All right. So basically, we are done with the investigation planning, and now this is almost all the knowledge we needed to know about the P6 basics of P6, because now what would be changed from now on? What would be changed? The only thing that could be changed after this would be the experiment. Only the experiment. Other than that, everything is cleared out.
Okay.
Uh, One thing I forgot to mention yesterday is the factors that were affecting the rate of transpiration. One of the factors that I totally forgot was the surface area of leaf. Surface area of leaf, as well as the location of stomata. Sometime it is included in uh, the surface area. What is the meaning of the surface area of the leaf? Remember, the surface area of leaf means that how much larger the leaf lamina. If leaf lamina means leaf blade. This is called as lamina. If leaf lamina is larger, then it certainly means that you are going to have a lot of transpiration. It means you have a large surface area. It indicates a large surface area. Then, like you may have seen the leaves which look like a needle. You may have seen the leaf that may look like this. So this has lesser surface area than this, and this has more surface area than this. So this is basically about the surface area of leaf. It would be directly proportional. Similarly, position of stomata. Remember that the stomata present at the upper epidermis, would cause a lot of transpiration, would cause a lot of transpiration compared to lower epidermis. That is why most of the plants have lower epidermis. That is why most plants have the stomata at lower surface. And even the desert plants shed their leaves to shed their leaves to remove stomata to remove stomata to avoid transpiration in driest season. In dry season. This was the point that I was forgetting yesterday. And then I told you a bit about the translocation. I told you that the glucose would be turned into would be joined with the fructose to sucrose. Sync. Yeah, no, I did finish it. Okay, this is yeah. One thing I was planning to tell you that sometimes the sinks become the sources. Sinks. What is a sink? Sink is the point where sucrose is translocated from the leaf. Translocated from the leaf. That is sink. But sometimes sink become the source. Suppose that initially what did you do? You had a leaf, you made the sucrose, and you transported the sucrose all the way to the roots. In roots, you had the onion. Now you uprooted the onion, and you grew it again. You grew it again. Now it would grow a new plant. It would grow the green onions. Green onions. Now, in that case, from where the energy came? The energy came because this was the sink initially, and leaf was the source. But now, once you have sown the sink into the soil, now it has become the source of the formation of these leaves, and leaves are now sink in this case until they are fully developed. Is that clear?
Um, yeah, that's clear.
Hold on. Okay, I would like you to read these two blue boxes. Be quick.
Okay.
Now these two, in these two, you can clearly see another in this particular leaf. You know that the plants that live in the water, they are supposed to be called as hydrophytes. The plants that live in the desert, they are called as xerophytes. Xerophytes. Now, xerophytes and the plants in our environment, in somewhat moderate environment. In the moderate environment, those plants are called as mesophytes. In the moderate environment. So if I clearly tell you, you have like upper surface to allows you to uptake. As far these plants are, since the leaf is supported by the water, a very thin cuticles in the water. Yeah. Now there are features that are very particular to the hydrophytes. Along with the book, you must also remember these features. Hydrophytes have air spaces in them, air spaces to support against water, against water, because their stems are poorly developed. Stems are poorly developed. Developed to support. Second and very important thing is their stomata are on upper surface to lose the water as they don't have lack of water, also to absorb carbon dioxide. These are the adaptations of hydrophytes. Then come the xerophytes. In the xerophytes, you would find xerophytes are for dry ones, dry plants. They have very thick cuticle, and you may have also read some of the features in the book. But most important feature that you have not seen is this one. Like you may have seen leaves collapse, stomata close to reduce heat absorption, evaporation, diffusion of water, leaf fall. Uh, then they have very less leaf, very less leaves they have. Uh, and as I have said, they would drop the leaves in drastic season. Drastic season. Similarly, there all this to avoid the transpiration, too much transpiration. They have sunken stomata. What is the meaning of sunken stomata? Sunken stomata means that this is the epidermis. Suppose this is the lower epidermis, and the stomata are just like present in the pit like this. Present in a pit like this. So they would not be able to lose the water. This is the stomata over here. And over inside it, you have mesophyll. So the carbon dioxide would be entering from here, but the this region would have a lot of water vapors. They have a lot of water vapors. Lot of water vapors, and these water vapors would create the humidity. And you know what would happen to transpiration when there is humidity?
When there is humidity,
Hmm. What would happen to the transpiration?
Um, we discussed this yesterday, right? So when there's
Yes,
more humidity, that decreases the rate of transpiration.
yes, it decreases the rate of run. Another very important thing you would find in the xerophytic plant is their, you can say, turned leaves, turned leaves, and in them you would find these hairs, these spine-like hairs. Why are they here? They would be trapping water vapors, and there is completely the benefit of trapping the water vapors. And there are also some of the there are also the stomata are sunken. Their stomata are sunken. So all of these things are in favor of xerophytic plant. These are called as adaptations of xerophytes. Understood?
Yep. Yep.
Great. Okay. So we are done with the transport completely. And then we need to jump to the transport in nutrition and transport in animals. Both of the chapters are quite large. Then after that, one more large chapter, one more, yes. Yeah, actually two more. Any question?
No.